Okay, my friends, this is very exciting from a research standpoint. This is Dr. Sabine Hassan. She's a doctor. She's in with all these other doctors at this big conference in 2024. It's called New Start. They want to open up medical research into the microbiome, which is not really being researched in a way it should be. Listen to what she has to say. Here's her story. Here it goes.
I kept collecting stools of patients and noticed that patients with severe COVID had a certain bacteria that was missing compared to people that were highly exposed to COVID but never got COVID. That bacteria is called bifidobacteria. Bifidobacteria is an important and key microbe for immunity. It represents your trillion-dollar industry of probiotics. In fact, when you turn the bottle and you see the ingredient, it says bifidobacteria. It is present in newborns. This is why your newborns did not get a problem from COVID at the beginning. And it is absent in old people. The process of aging is loss of bifidobacteria. We published this paper, The Lost Microbes of COVID-19. It took 8 months to publish. If you follow the bifidobacteria like I did, you will notice, and we did notice anyways, that vitamin C actually increases bifidobacteria. This is why vitamin C is important when you take care of viruses, and you've all experienced taking vitamin C for a cold. Well, we published this data where we showed vitamin C, if we give it to patients before and after, it increased the bifidobacteria. Ivermectin was also an interesting drug because ivermectin, we noticed, also increased the bifidobacteria within 24 hours of taking it. Why ivermectin? If you look at what ivermectin is, it is a fermented product of a bacteria that is similar to bifidobacteria. In fact, they're in the same continent of microbes. They're like sisters, brothers in the microbiome. So I published, I knew that ivermectin increased bifidobacteria, but I said, I can't go out there and start publishing that. That's going to be too controversial. So I published a hypothesis that maybe what I was observing on the front line treating patients with COVID, noticing that their oxygen saturation was increasing from ivermectin, was basically maybe ivermectin increased bifidobacteria. The hypothesis on ivermectin was the most read hypothesis in the pandemic and was retracted after 8 months of being on. When we cannot make a hypothesis, this is not science.
Okay, my friends, this is very interesting. This is Dr. Sabine Hassan. She did some research on the microbiome, and I think I have some things to add to it. So we're going to look at this. Now, this is research only. There's no medical advice whatsoever, will I ever give. So you can see the research, do your own due diligence, look up and see if I'm telling the truth or if I'm wrong or whatever. But I go by people that really do research, and she is a researcher. She's not just a, take 2 of these and you'll be better in the morning type of person. She wants to know what causes these issues. What is the cause of all these people's concerns about vaccines and about, the microbiome and all this stuff? Well, what can we do to test it? This is where I fall in. I'm a researcher. I'm a material researcher. I like to see evidence. I like to see tests. I like to see results. I don't like to make up guesses. Although I do follow the research, and you have to make assumptions then and try to figure out if they're right or wrong. Don't just make them and say, that's just the end of it. I just figured this out. It was all done. And that's what happens now in medicine. Although I see this is changing, and you'll see what she says, because the doctors had no clue about this. This is only a year ago this video came out, and I think she's still struggling to get people to pay attention. That's the problem. You can have all the evidence that you could possibly want. People just don't pay attention, and especially the ones that are already invested in some form of way of doing things that they're not going to change, even though they should understand that they should change, or that maybe is a possibility of a change. Let's study these things. It's better to stay away from them for their well-being, let's say, because they're going to be hurt by doing this research. Nobody's going to pay attention to them if they do research outside of their realm. Trust me. That's what happens. They say this guy's an idiot or she's an idiot, but she's a doctor and this is her realm and this is her area, but they still wouldn't help her. She had to do all this herself. Now I'm going to show you what she has to say. Very, very compelling stuff. And I think it has a lot to do with the future of medicine, basically. The microbiome now and the virome, which is nobody's understanding, is the biggest thing in medicine right now. They call it the microbiome. I call it the microbiome. You call it any way you want, but it's the biology that's in your gut. And there's not only that biology, which is bacteria, there is also viruses, and I call that the virome. So biome, call it whatever you want. But that's the bacteria part. Then you got the virus part. And I'm going to split that off into the virome. They have two totally separate jobs to do. And I'll explain to you what those two jobs are. And I will show you evidence of material research that points to what those jobs are, and if they are interrupted, what would be the outcome? What is the result? She knows because she's been tracking it and taking samples prior to certain types of things in your microbiome. What lives there, and then after some kind of medical intervention, let's say vaccine or antibiotics or whatever, what's your microbiome after that, and how long does that last? And does it repair itself if it is damaged? All of these things we're going to talk about. Okay, so what are we talking about primarily? We're talking about bacteria. We're going to look close at that. Where do the bacteria live? They're all over your skin, they're in your mouth, they're in your eyeballs, in your ears, and everywhere. They're everywhere on you and in you. There's more of bacteria in you than there is you in you. So, and every one of them has a job to do, and they create ribosomes, which I will show you in a minute. Now, where do they live? Primarily, they live in your gut, but they also travel through this fluid-filled highway. All right, this is the interstitium. This is that newfound organ. That's the tubular fluid-filled highway that I wrote the paper on 2015. And all of these enzymes and bacteria flow through there, but they originally come from the gut. Now, but they go to every organ and everywhere else. And you say, oh, how do I know that? Well, I didn't know that, except I was talking to sports injury doctors over in Germany. And one of them said to me, he said, you know, we're finding tattoo ink down around in the organs and in the membrane, you know, in the tendons and everywhere else. I said, tattoo ink? Well, how'd it get down there? And they say, well, they don't know. But this one certain type of tattoo ink, I don't know, it was black or something, I can't remember. It was one type of, they were finding it in the tissues of the membranes, because this was like the Fascia Institute in Germany. So it was a big place, and this guy was very accomplished, and he was one of the only people in the world who knew anything about it. And I guess they were looking at it to see how it interwove, and, you know, primarily for sports injuries, I believe. And, you know, aging, fascia is a real thing. You want to keep that stuff moving. Otherwise, it glues up. It literally turns hard and glues up. And I talked to the fascia, the guys that do the autopsies, very, very accomplished people. And they said the same thing. They said when they cut open some of these autopsies, they know if that guy was stiff or not, how tight the fascia was. And very, very, very slow stretching in all the different ranges of movement are pretty important, especially when you get to an elderly age. Now, so we know we need bacteria in here. We know we need enzymes. What do they do? What do they look like? Where do they live? Well, we know they live somewhere. Well, they live in your entire body. Just forget about that, because they're everywhere in your body. As far as I know, they're in your bloodstream. They're in your lymph, primarily in your lymph, which is primarily that layer. That's your lymph system, almost. You know, it's everywhere in your body, but that's where it gathers. And when I say gathers, well, what do I mean by gathering? Don't forget, these are bags of membranes that coat something, like a lung, all right, like whatever, like these breasts of muscle. You see that? That's tendon. I mean, let me scoot right in there. You see that? That's fascia. Now, you got a little really gnarly thing that you chew on it, you throw it away. That's the latch. Now, from that latch, you have the bags. These are completely encased. You see them, they're ripped open, but when that thing was alive, all of that fabric coated these things, just like a rubber bags, just like they were in 2 rubber bags, completely encased in rubber bags, totally separated from every other molecule that was around them, 100% separated in their own little bags. And then they had to come together, so the fluids that flowed through these membranes in these bags, they were nothing more than a whole batch of fluid-filled highways. Where does that fluid go? It's got to go out through that little tiny drain. And I can show you that right here. This is what mud fossils are so good for. That is the drain right there. And I can actually see the two outlets. They're like little tubes coming out of there that will bring that fluids that are in here into the next organ or into the body cavity, wherever that attaches to. Now, what does that mean? Well, we know we're going to have enzymes and fluids in here. Are they special? Are they special because this is muscle? I would say they are. You wouldn't have the same kind of enzymes and fluids around your kidneys, around your heart. Well, maybe around your heart because it's a muscle as well. But you wouldn't have that around your pancreas or your liver. There'd be some kind of different chemistry in that fluid-filled bag highway. And then that would have to drain into something else. Now, would you drain that same fluid, or would it be filtered there? Is that a filtering device? I don't know. These are things I have no idea about. And nobody does, because nobody even knows about this. I named that the Spurlock. And I've talked to top anatomists in the world. They don't know about this. This latches, in this case, muscle. In other cases, it would be a lung. Or your heart. Everything is locked in somewhere so that it doesn't get ripped and moved around. And then everything is coated with its own membrane fluids. And those fluids are not going to be the same in every different area in the body. So how do they change? How do they filter them out of here? This is starting to play a lot of games in my head. It gets deeper and deeper as you go along. And it's just, there's nothing you can do about this. You got to keep thinking and thinking and thinking. Right now, they know the bacteria causes it, but when we get way down here, maybe you don't even need to get this deep, but I do. I can't help myself. What is it that in this membrane that's protecting you? Is there some special bacteria in a specific membrane? And I think there probably is. And you have thousands and thousands and thousands of different bacteria. And every one of them creates its own specific ribosome, which is that ball. And then they just float around. They're free floaters, they call it. They float around your body, go anywhere they want, and they collect inside the cells. And when they want it to do a job of specific instantaneous chemistry, because this is an enzyme, they go, and all of a sudden, all kinds of things happen. And they happen due to this half-life and decay and daughter isotopes of the primary players in this, the primary molecules. When you hit them with enzymes, the molecules literally do something that's called nucleosynthesis. the nucleus changes and they get synthesized into something else. Sometimes they go up from being a small atom down here, they jump up, and they can do it because of these enzymes. During the half-life, so they can go and move up, or they can move down. But these are the things that do it. Without the enzymes, you're done. And the enzymes, well, let me just show you something. Okay, my claim, as I said, was the ribosomes turn into these exotic enzymes, and then it's done instantaneously. And when I say instantaneously, check this out. This is the reactor, which is they don't get used up. They go and they change things so fast it's beyond the belief of the human mind. And this is all done by these tiny little enzymes. This appears to be accurate. It says the enzymes are remarkable biological catalysts, significantly speed up chemical reactions. They can accelerate the reaction by factors of millions or even billions compared to the rate at which the reactions would occur without them. To give a bit more detail, the speed up factor of an enzyme is typically quantified by the catalytic efficiency of the enzyme, which is a combination of two key parameters: the turnover number, the number of substrate molecules converted. So it's changing things in a certain turnover per product, per enzyme molecule, per unit of time, when the enzyme is fully saturated with the substrate. Here's the constant. Look at this, K1 to 2 to all that stuff happens within like a second. All of these are turnovers. What does it mean? You look at it and you say, oh, it's just too confusing. No, it's not confusing. It's not confusing at all. Take your time and think. The catalytic efficiency, that means how quick it catalyzes, how quick that enzyme reacts. There's two parameters, two different things involved in this. The turnover number is from number 1 to 2 to 3 to 4 to 5. They're making a molecule change from this molecule to the next molecule to the next molecule by this catalyst, by converting this. So that's the turnover number. And then the number of substrate molecules, which is what it's growing on, it's moving through that. How much of that stuff, those molecules, are converted to the products per enzyme molecule. So if you have one of these enzyme molecules, how much are you going to create of product? And in what amount of time? If the enzyme is fully saturated with substrate, look at what it creates. All that stuff is happening because of one of these. They just, and what is it doing? It's doing half-lives of all the molecules that it interacts with. People never consider that. Isotopes have a bazillion different half-lives because the standard model is wrong. The standard model is not correct. There's so many different particles in one proton. There's at least 1823. There could be up to 1840, somewhere around there. In a ball like that is a proton. It's not one big ball like this is a proton. No. It's 1820. Let's say I go with 1823, 1839, somewhere in that area of dipoles. It's the same as this, but they get stable at that. They go, and then they lock in. And at that unit, it appears to be something in the frequency of somewhere causes these to become stable in quantities which associate to these atomic elements. But all these are is bigger snowballs. All these are is bigger snowballs. And they have certain characteristics associated to them depending upon how big they are. Some of them are gases. And I can't account for all of the characteristics, but I do know some of them, like, well, anyway, there's a lot to it. This gets very deep, and it's hard to understand because the periodic table is not correct. It has to be changed a little bit to accommodate for the quantity of particles that's inside of every other particle. All right, so like I say, this gets deep. You can't just go for one little piece and everything's fine. Oh yeah, I know everything about it because I know X. Well, you got to know A to Z, my friends, or X means very little. All right, my friends, as you know, for many years now, I realized that the microbiome is the source of health, basically. And I just do research. I don't recommend anything specific, but the bacteria, even Hippocrates a couple thousand years ago said, all disease starts in the gut. Is that right? Well, maybe not. That's what I do. I do research. So this is a doctor. She is a doctor, and she realized also that the microbiome had extreme implications. So she started doing fecal swabs prior and after to anything medically intervention, antibiotics, vaccines, whatever it was. And she found out there was some significant differences. I'm going to let her describe her experiences. I'm going to cut in here and there, because I also had virtually the same experiences she's talking about. And I support everything she says, and she's supporting everything I say. So let's get into it. Okay, my friends, I'm just going to let this play through once, and then I am going to address her main statements that she talks about in we'll replay it, probably not in its entirety, but we will replay it, and I will address, because I have so much more to add to this. And I'm a material guy. I want to talk about the enzymes that are produced by the bacteria that are being killed. What do the enzymes do? Where do they live? How do they react? What do they produce? How are they actually programmed? All of this stuff is my realm, is a little deeper. She's getting to the point where they realize the bacteria is the issue. Now, if you replace it, well, that's fine. But I like to know, why is that bacteria the issue in the first place? Why is it there? What does it do? What's its interaction? How does it work in the membranes, in the fluid-filled highway, in the fascia? These are the things that I actually discovered. The fascia was the fluid-filled membrane, and all of these things reside there. I think I have a ton to add to this, but I want you to hear what she has to say first, and then after that, I will make my statements. Here goes.
I said, well, you know what, since the agenda is the vaccine, well, let's look at what the vaccine's doing in the microbiome. And that's at the same time as they started rolling these vaccines, I started enrolling doctors that basically were getting vaccinated. And I'm like, can I get your stools before and after you get vaccinated? And sure enough, people would come to me and they're like, hey, I heard you're testing the microbiome. I want to have my baseline, because just in case I change, I want to know what microbes I had before. So I'm like, yeah, happy to do it. So, this study, that was, again, something I undertook myself and paid for myself. By the way, we applied for grants and all that, waste of time, waste of money. So I basically just dumped my money into this trial. So the first 4 patients, I started noticing a month later, the bifidobacteria, this important microbe, is dropping in patients pre and post vaccination. So then I started asking myself, wait a minute, what's going on here? I mean, is it creating a bifidophage? Because this is precision medicine. This is forensics of the gut, right? You've got your microbiome this way before, and you've got it after, and it's the same patient, and only a certain group of microbes are getting killed. You got to pay attention. So then, 10, 20, 30, 34 patients later, we're seeing this, killing of the bifidobacteria. And so I wasn't going to, first of all, there's no way I was going to publish this, because nobody would have taken that. So I decided to submit it to the American College of Gastro as a presentation, as a poster. It got accepted at the American College of Gastro as a poster, and then it won the Best Research Award as a poster. So all my colleagues called me and said, hey, I saw your data. That's incredible. How do you think this is happening? The vaccine's supposed to be, improving your immunity. And we all know bifidobacteria is a huge part of immunity. How do you think it's happening? So then I said, I think it's creating a bacteriophage or bifidophage. And what we noticed is in 4 patients that we followed, which were amazing shape, we followed them for 90 days, and then next thing you know, their bifidobacteria dropped to 0, from 1 million to 0. So it kept persisting. So there was a persistence in the damage, and not only 90 days, but 6 months, 9 months later. So that was the thing that started making me panic. And then as we were looking at the microbiome of newborns, to mothers who were breastfeeding, we started noticing that there's no bifidobacteria in those newborns. So we asked ourselves, well, I mean, because newborns are supposed to have a ton of bifidobacteria, right? 90% of the microbiome of babies is bifidobacteria. So we said, well, how come these babies born to moms that are breastfeeding that were vaccinated have zero bifidobacteria? Is the spike protein going to the breast milk, into the baby's gut, and killing whatever the baby's trying to build? And so because I was doing work on autism, and I noticed, one of the commonality and common findings of autistic children is that loss of bifidobacteria, I said to myself, maybe that's how it happens, right? Maybe you're killing off your bifidobacteria, and then 2 years down the road, your kid stops talking. So this whole, it was serendipitous, really. I mean, this whole discovery, because, understanding what the microbiome looks like in Alzheimer's, in old people, in overweight patients, in Parkinson's, and, kids, healthy kids, kind of brought it all together for me. So I had a different vision than everybody else. I was looking at how to treat the virus, knowing something nobody knew. And that was really, it was kind of epic. But at the same time, I knew that, hey, they can't prove me wrong. And if they are going to prove me wrong, well, let's see why. But definitely this needs to be looked at, because the microbiome has such an importance in neurological problems, in cancer, so in Crohn's, in Lyme. So, we just published 2 posters that were presented at the same college where basically loss of bifidobacteria was noted in Crohn's patients and in Lyme patients. So bifidobacteria has a big role in disease, in my opinion, and so we have to pay attention to what's killing it.
So, I mean, your hypothesis right now is that it's the spike, whether the spike is coming from the virus or whether it's coming from the vaccine, that this is actually causing this, it's killing the bifidobacteria, which is creating a problem.
Correct.
And this is what you're hoping, some serious research is done around.
Yes.
Yeah.
So, and we are doing something to try to prove that. So it's coming along. Obviously this is research and it's slow developing. Research is not fast. It's one thing proves another, and opens the door to another. I'll give you an example of research during this pandemic. One of the things that I did myself was kill my bifidobacteria, because I wanted to see, well, what increases the bifidobacteria, right? So I was the guinea pig. The whole pandemic. And, first to see, if I'm exposed to it, etc. And it basically, what I realized, one of the things, I was drinking a ton of kefir, and my bifido is not increasing. I'm not really good at taking my vitamins, right? Because I'm stressed and I'm busy and I'm not a pill taker. But essentially, I kind of let it die, and I started testing my kefir, and I looked, because we passed it through the machine, and we noticed that the kefir didn't have bifidobacteria in there, even though it says bifidobacteria. So I went to Whole Foods and Ralph. This is research, because here I am, I've just killed my bifido. I'm trying to boost it. Why isn't it increasing? So of course it opens a new research. And I went to Ralph's and the Whole Foods, picked up all the products that said bifidobacteria in the back. And we took 23 products. One of them was a $27 tonic water that had bifidobacteria in there. So I said, well, this gotta be bifido, because $27. I mean, come on. Anyway, so we shook them, we took the sample. Only 3 of them
had
bifidobacteria.
Unbelievable.
So so you become an aware customer, right? So this is what research is all about. Research is about, why isn't this happening? Why isn't it? Let me figure out why this is not happening. And of course, my kefir didn't have bifidobacteria. How am I going to increase my bifido if it doesn't have bifido, right? So then I started doing my own stuff, the fermented foods and all the stuff that I know how to do. And basically, I'm happy to say I'm back to my normal bifidobacteria. But this is what research is about. It's finding one thing and then opening up a new science, a new research to kind of discover. And so that was fascinating.
That was great. Okay, we are going to be talking about the research being done by Dr. Sabine Hassan, who is a specialist in gastroenterology, exactly where all of the microbiome is, which is in internal medicine and hepatology. Dr. Hassan has used her medical expertise in many regards over the last 2 decades. Now she is looking deeply into the microbiome and getting started because it has been overlooked, and it's still overlooked in most cases, to its full potential. Let me put it that way. It's now becoming incorporated in, as they realize it's an absolutely essential part of the immune system, but still there's a lot of vagueness to exactly how it works. I am a material guy. I want to know what happens when the bacteria dies, what misses, and what was the part that's missing, what was it supposed to do, and how did it do what it was supposed to do, and what did it need to work with that. There's all kinds of complications once you get to the fact of knowing the stuff is missing, so you get sick. Well, that's good. Put some more in, that's fine. And really, that's probably all you need to know, but I can't stop there. I can't. I want to know why. If it's missing, what should it have done? The whole process, right down. And I think I do now fully understand it, as much as anybody does. And I would like to be a part of this whole discussion. And that's what we're trying to do, is get a bacterial database, which is the standard of everybody. Like she's saying, this particular bacteria is missing. And that's what people are getting sick, literally sick because of it. And I know I have evidence to show that what she's saying is almost just absolutely perfectly correct, is my opinion as a researcher, not as a doctor, as a researcher. And again, no medical advice whatsoever, but literally everything in your body is related to bacteria, which create enzymes. Enzymes do the chemistry so fast that you have to have them, otherwise the chemistry will never be done. The enzyme will do more chemistry in a quarter of a second than would happen in over a year in many cases if you didn't have the enzyme. And if you don't have the enzyme, it means you didn't have the bacteria, because the bacteria create the enzyme. So again, I have a lot to add to this, and I know how the membranes work, and I know really most of it now, because I've studied it very, very deeply, understanding about the bacteria creating the enzymes being literally created from ribosomes, and the ribosomes are so small that they can go through the blood-brain barrier. There's all kinds of implications here, understanding the bacteria are the source of the ribosomes, which is the source of the enzymes, which is the source of the chemistry, which is the source of every single thing that's done in your body, 100%. All right, so let's get started. All right. This is not bragging, it's just I have to establish I have some credentials here. This is an enzyme, and they say, oh yeah, you have to have these, they're catalysts. Well, what does that mean? A catalyst means if you don't have this, the chemistry that has to happen to make your body run could never happen. It just can't happen, because the chemistry that has to make your body run has to happen instantaneously. So you have to have a carrot come into you, and almost instantaneously it has to do whatever it does, make some bone or energy or whatever it does. And this is what does it. When this hits other things, it works millions, even up to 1 trillion times faster than if the other thing just had to change by itself. It would change by itself, but this just says, done. Nobody knows how these work. You say, how does that happen? And they don't even get used up. I call it click chemistry. That big ball of magnetism, and that's all it is. It's just like this. I got one around here somewhere. I'll show you in a minute. It's a big ball of magnetism. And it goes click, 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 and it kills all these other things. As soon as it hits it, they're done. And they happen instantaneously, like I say, in a quarter of a second, or in a second, let's say. That thing there could make more chemistry happen than would happen in a year or more. Without these, you have to have them. So these are only come from bacteria. That's where I go. I go down to, why is the bacteria an issue? We know if it's not there, we got a problem. But why, if it isn't there, what is the problem? What does it cause? This little molecule is missing. So you can't break down or create whatever you have to create. These are like enzymes or they're proteins. They go both ways. The enzymes break things, the proteins build things. And we're going to go into that. But before we do that, I just want to show you. I did all this stuff. I did all the chemistry, the bonding, the this, the this, the that, the that. And then I went into the, thermochemistry and all the different, I did every single thing you could do in all the light research and photons and chemistry, bonding chemistry and periodicity. This goes back a long time ago, but I'm telling you, I did it all. I can remember it all too, basically, because when I get into something, I get kind of deep. That's the way I am. All right, so, and that's my point. I'm not bragging, like I say. If it's true, it's true. If it's true, it ain't bragging. All right, so here's the ball right here. This is what we got. You see that? I'm going to show you what a ribosome is. It's a little tiny ball. So tiny you could fit 1 million of them inside of one single human cell. 1 million of these in one cell. But once they open up, they go and they get huge like this. So the key here is in the blood-brain barrier, you can only allow the tiniest things in. These are like pills. These are like chemistry pills. So they can go in through your blood-brain barrier, no problem. And when they get inside, they're just sitting around waiting. And when they get energized, and they kill something or build something, but that would never be allowed to get into your blood-brain barrier. We got a lot of talking to do about the effect of missing bacteria. I know the missing bacteria, if you replace it, you should be okay. But first of all, we need a database of everybody's standard sort of bacteria. And it may even depend on your race, your sex, your age, all kinds of things, where you live, what kind of water you drink. There's going to be a lot to think about here. But the end result is your microbiome, your microvirome, the inside of your guts, what is being produced in there. That is digestion, metabolism, breaking things down and building them back up into your body. If you don't have the right little guys in there, it's not going to happen. These are the most important things in your whole body is bacteria. All right, this is my paper from 2015 about my fossils, and I'm going to show you the fossilization of the fascia and how the fascia protected the things to fossilize. And we're going to go in a microscope in a second. But this is from 2015, and I could see these things, and I questioned some people about them, and nobody really understood it. And this is what I wrote in this paper, 2015. I said the fascia was thought to be mere packing material, virtually inert. It has long been ignored as a subject of research. They just thought it was just nothing. Recently, however, there has been new equipment that allows viewing of the fascia as it does its job. They can see it moving around and all that, and I could see the things, and I was looking at this too. And from the experiments, it was totally unexpected results. Fascia is a tubular, fluid-filled fabric network. And you can see this. It's not a question about it. Now, how does it react in the whole body as a system? I called it a fluid-filled tubular network. Now, this is 2015, so I had about 5 years into this already. And like I say, when I first started, they called fascia fuzz. And the first guy I talked to who was very, very accomplished, he does presentations all over the world and tours and everything, and he said, oh, that's just extra stuff, and it's just packing material, it's fuzz. And he actually had the video called The Fuzz. And then a couple of years later, after we talked a little bit, I saw another video he had up there reconsidering the fuzz. Now, he's never endorsed anything of mine, and I don't want to use his name because everybody seems to be associated with me, gets in hot water with the big shots. So I went through this whole thing about how the fascia worked and in the body, and at the same time I could understand how the transition of molecules was going through the membranes and all that other stuff. And then we come into this enzymes and bacteria that live in that fluid-filled highway. And there was more video that came out showing the interactions inside and the ribosomes and then squirting right out into the fluid-filled highway. You could actually see it happening. So there was so much research just kept piling up and piling up. And I kept doing more and more videos about it, but just like this woman is showing, she's a doctor, and it took 8 months to even let them put the paper out. And then when they put the paper out, it was #1, and then they made them retract it. This is the kind of thing we're up against. I can't understand it. We're supposed to be trying to understand things. Instead, it seems to be the opposite. I just don't know what to think anymore. I really don't. Okay, so what am I hoping to see out of this research? Well, a universal database. And what does that mean? The bacteria that's in your body, you've got a zillion different species, and there's certain quantities of them you should have. You shouldn't have too many, too little, but you should have good ones and bad ones. You have to have them both. And I'm serious, because you have flesh-eating bacteria in you because you've got dead flesh in you right now. And something's got to clean it up. Now, you don't want them eating the good stuff. So something's going on with bacteria. They can work with each other. But one of them gets out of control, it's all over. Now, we need to know all the bacteria. Well, as much as you can know, that's what that means. As much bacteria as we can figure out, and how many species there are, and the quantities that are in you. And we're just going to use the fecal swab. We need to have a huge database, huge, of everywhere in the world, because the water you drink where you are, the air, the customs, everything else, that's going to all play a part on what your microbiome is. And you may have to have a different microbiome than I have for the American diet. But we'd like to know. There's only one way to know, is to accumulate the data. And this should be very, very, very simple. So we need to know a huge, and then filter it down into the regional and everything else. How illnesses and, our age and your, even, like your sex, even race and, everything about you. As much as you want to put in there, that'll be in your database, and it'll be in the worldwide database. Now, but we'd have to develop a really simple, cheap kit for a little poop swab, something, and some way of preserving it, or whatever. That's something chemists do. They should be able to. I know they're doing it right now. They're harvesting this stuff, but sometimes they say you need to have it there within 4 or 5 hours for it to be viable. This is something we have to look into, because I don't take anybody's word for anything anymore. You're going to have it 5 hours. No, you go 24 hours. 48 hours. Some of them they have the dried out, desiccated pills. They're just dried out, and they put them in capsules. Now, what's the story with that? They say those work. Well, who knows? But anyway, we need a simple questionnaire. Simple. Check this, check 1 to 10 symptoms, anything you want. But a very, very detailed questionnaire would be best, but at least you get some of the data. And we develop the patterns of people that are sick versus not sick, and the different types of microbiomes in literally different areas where they live. Because you're going to be affected by something that's going to be different where you live. Even diets, the water will be different, the air is different, dust and all that stuff is going to be different. Well, does that affect it? I don't know. Is your microbiome supposed to be flat out just like this no matter where you live? I don't know. Nobody knows. I don't think. I did see something. They said there was a worldwide database, but I don't know how you'd get into it, or that's kind of outside of my realm. I do the research, and this is what I want to see happen. I want to see a worldwide, easily to access database that say, I want a kit sent to me, and I want to be part of this thing. Boom, done. Then, as the thing grows, we can find out. But right now, it does appear that she's onto something with this particular research that she's doing right now. I'm not going to make any claims about anything, but it appears that this particular type of bacteria is extremely important for your digestive system primarily. You know, because that is where all the stuff happens in your digestive system. So let's look into this a little deeper. I just want to show you for now what the bacteria does and these enzymes that they create. They just don't understand it. They know now that the bacteria, if it's dead, you're done. All right, and so you're going to get worse, and the COVID's going to be worse, and all these things will be worse if you don't have that bacteria. So what does that bacteria do? The bacteria is single cell, little tiny. These are so tiny, the ribosomes that they create are so tiny that they can go anywhere in your body, right through the blood-brain barrier, anywhere they want. They're like this. And they squirt them out all day long, according to what the program of their own DNA is. Don't forget, this is single cell. This is phenomenally unbelievable how intricate the human body is on this micro scale. Now, these squirt out. Don't forget, one single ball like that of ribosome, you could have 1 million in a single cell in a human body. 1 million of them. They're that small. That's why they go through the blood-brain barrier. Now, what happens when they open up? First of all, why would they open up? Well, let's think about that. All right, this is a bit of a mystery. There's a trigger that opens up these balls. What that trigger is, they think there might be some little hairs or something on here, or somebody squirts another enzyme onto this and makes it pop open, whatever. Somehow, the trigger makes it pop open like this. The sheath comes off, and this pops out like this. And they are exotic, these enzymes, or proteins. Proteins are more like this. They're like building blocks. Enzymes are like all over the place. They're just, there's so many different bacteria, and every one of them has an exact. And when I say exact, I mean exact. If it's not exactly that, it's not going to work. That's called denatured. All right, so if that protein is like this, supposed to be, and this is like that, you're done. Not going to work. So you have to have the perfect protein now, or I mean a perfect enzyme. And that requires the bacteria. And it requires the bacteria probably to be healthy, I would imagine. I never thought about it, but they may be able to make defective ribosomes. I don't know. Maybe that's part of where viruses come from. There's so much detail down here that nobody's ever really considered. Like they say, they just now getting to the point where they realize that bacteria, missing bacteria, is the cause of some serious issues. And like I say, I want to know why that missing bacteria creates that issue. And here is the problem. You don't have that enzyme. Don't have that enzyme, you're not going to get the chemistry. You don't have the chemistry, you're not going to build something, or you're not going to break something. Both of those things are required for life. I know I drone on about things, but this is so important you understand. That ball turns into this. The ball is the only little thing that can get through your blood-brain barrier. You try to make that go through the blood-brain barrier, it won't go. That's why it's there, so they won't let things in that shouldn't come in, because an enzyme is very, very, very active chemistry. And then the protein is the same thing. They build up plaques and all kinds of, they keep it out of there. But the ribosomes should be able to go wherever they're supposed to go. So anyway, that's that part. You know, I always go a little too deep, but I'm going to do it anyway. And I'm going to try to breeze quick. There's amino acids, there's 20 of them. They come out of these polypeptide chains being squirted out inside that bacteria, and they turn into a ball called a ribosome, which programs messenger RNA. And then the messenger RNA, this is the whole sequence at once. The bacteria create the ribosome using these polypeptide chains. In here, using transferase, which is an enzyme, and it transfers into the messenger RNA, and then using polymerase, another enzyme, it breaks into our DNA. That's as much as you have to know. You want to look it up, figure it out. Now here's where I think they're kind of important. vitamin D and C, specifically they're talking about, and some other vitamins can pass through the blood-brain barrier. So they're going to be in amongst the places where most chemistry can't get to. So they are very, very, very tiny, these vitamins. That's why they can get in there. And I believe the vitamins are necessary to work with the enzymes. They're like an intermediary. I haven't really worked this out yet, but I can tell you what, when I did look at the chemistry, vitamin B, I think, is probably too big to get through. Vitamin B12 that's a big molecule. Hold on, let me just see something. Oh, this is going to be cool. Wait till you see this. This is chicken breast. I think I showed you this before, maybe not. But this, you see this really strappy looking stuff, and you get this on a chicken, you maybe have had it, I don't know if you're a vegan or not. Now, that's the little lock, and that's the latch, and all that filtered fluids have to come out of that bag somehow, and they go to the next place where they lock in. Now, I'm going to show you that place in my microscope. I have, and they're mud fossils, and it's going to be this rock right here, this rock, you see it? That's the fascia. That's the meat of the rock. It's exactly identical to here. That's how they latch. This is ripped. You see this? This is a sports injury. This is where I get a lot of my information is from when you're looking at these biology, these body parts, and they're ripped and broken. And then I can see them. Now mine are not ripped and broken. They're just boiled and disassociated from the rest of the body. You see that? Look at this thing. I got it here in the microscope. This is tough as hell, that latch. And I discovered something that I never knew before. There's a little round bottom. I'm going to show you in a second. Now, again, they latch together so that you can pull against one and the other. Wherever you have a body chunk of muscle, it has to lock in somewhere, and that's the lock. I call it the spur lock because I discovered it. All right, I have that rock right here up in the microscope, right up here. All right, now, that's that latch. All right, it comes back right here and latches right here. This is the toughest, gnarliest stuff right here. And that locks in to in this case, it was a muscle. I have other ones that are lungs. They're all basically the same. Now, all of that stuff from the fluids that were in this bag have to filter through there to the next place. And there's two little dots right here. These two little dots, I'll show you in a microscope. That's where they filter through. Hold on. Well, I'll have to refocus, but that's where they focus through. Now, then there's also a spot on the bottom. Let me do a little freehanding, so this is going to be a little shaky. Here goes. All right, now, don't forget, this whole thing here is muscle. Well, what's that big white thing doing there? The latch is hanging over. That's not attached to this. That was the attachment to to the bone or wherever. You see that round thing? Take your time and look at that carefully. Let me see if I can focus in a little better. Well, it's pretty easy to see. This is meat and that is muscle, or an anchor of some sort, which is the same sort of connective tissue. Now, so this came over and this latched on top. It's like a clip. It looks like it to me. But there's no question what this is. It's meat, and that is the latch. And I have so many of these, it's unbelievable. Every single body part has that latch. If you get the right place. Sometimes they break off or you don't see it. But when I see them in the lungs specifically, they're all over the place. Now, this spot right here, let's do a little more free handing. Hold on. Well, you know what? I can turn. I have a nice little thing here. Hold on. You see the difference in coloration? You see this? I'm going to come down here and show you this. These are the two spots. We're going to get a little closer. And that's what drains this. Now, could that affect? I don't know what to think at this point. But I do know these organs have a completely, totally encased, separate membrane around them. However, they feed into the entire body system, the fluid-filled highway. Is this a different fluid? I don't know. Maybe lymph is the same, because I think this is primarily lymph. I think lymph might be the same through your entire body. However, I don't know. I wouldn't, at first thought, I said, no, it's impossible. There has to be some different chemistry, and especially where you got, say, your liver and you got, your kidneys and, all that kind of stuff. There got to be some kind of different chemistry in that membrane, I would think, but I don't know. I don't know if anybody's done the research to determine whether the membranes themselves have different chemistry within them. I don't think it would be a big deal to do that. But I don't know. But anyway, that's the same as the rock that I showed you down here. Now, let's come in and look at those two spots. Let me focus in and then I'll start it back up. Hold on. All right. I know people are going to see all kinds of things in here, which is not what the things that they're going to see. That was that attachment to the muscle or to whatever. And this is the flap that comes down. These two spots right here are not eyeballs or anything like that. This is that chunk of meat. These two things are where that fluid flows from one to the other. That's the only thing I can think. That's the only thing I can think. And don't forget, now this is the very tip. Now I'm going to move this thing up slowly so you don't lose track of it. No, I'm not playing any games. I come way up here, it's way out of focus, but then I'll bring it back. All right, now I got to bring it back into focus. Hold on, take it easy. Oh, you can see the two white dots, and that's the thing that runs down there, the whole latch. You see it? I mean, I know it does look like somebody's face or something. That looks like another face. People see so many things and think. You have to understand what it is. Just because something looks like something doesn't mean it. But when you know what you're looking at, it does mean it. And I know what I'm looking at, and that right there is a spurlock. And as you follow it back, right up there, that is the gnarliest, gnarly stuff you ever saw. Yeah, right there. It's really, really tough. And that's what anchors you so that it doesn't rip out, so it has to be tough. And but that right there is where the fluids drain back to the next organ. There's almost no question about it. What that means, I don't know. Maybe it means nothing. Maybe it's just a fluid-filled highway. Is there a difference in those? Let me come up pretty close. Hold on, stay with me. Wow, this is very interesting. I have always looked at it kind of far off and see this one and this one, and those are the two little spots. But look at this. That indicates to me like a vein-ish sort of thing. The real white, look how white that is. That would be like an artery sort of thing. Now, I know that they're not a vein and artery, I don't think, but they might be, I don't know. How does that fluid flow then? Maybe this isn't where the fluid flows. Maybe this is just a vein and artery that supply that. I don't know. At this point, that's a new revelation. Maybe I don't know what. But there's something going on here. You see, whenever you see something that's different than something else, there's something going on. This is a real bloody area. That, I would say, looks to me like it's from a vein. And that looks like red bloodish stuff, and that looks like black bloodish stuff. Now, is that what it is? I don't know. I do not know. Let me come up a little bit. Let's come up just a hair. Let me turn the lights down a little bit. Boy, I don't know. I've always thought that was the ones. But that, I still do. I got to be honest with you. When you step off a little bit, sometimes you can see better than being right on top of something. I got to go with these two. But it looks like there's a vein down there. Maybe there's an artery over there. I don't know. These are things, and now I'm looking at it, they very well could be. You see that flap coming right down the middle? Maybe this comes straight across, and this comes straight across. Because you can actually see that may very well be the case. Something's going on there. Anyway, it gets very deep, and you have to spend a lot of time looking at it, because you may look at this for 5 minutes, and the next time you look at it, it's totally different. And that's the case in here. I really looked at this thing a lot. I thought I knew everything about it. Maybe I don't. And even now I'm looking at it. I'm seeing this fracture zone here, and there's another exactly almost identical over here. This is almost a piece on its own. And I would say that looks like red blood, and that looks like black blood, and this looks like it follows the same blackness, and this follows the same redness. And that's how black and red veins and arteries run. They run on their own sides most of the time. And all of that is, that's fascia. And that's all really strappy stuff. Right there is the investment. That right there. That's the key. Let me see if I can get a good shot of that. That's where it locks in one body part to the other. And that is some tough stuff. And then it rolls right off into the fascia and the bloody area down here. But back up here where that latch is, that's where it's really, really locked in. It has to be, otherwise you rip your body. You think of a football player. When they get hit, all their body parts are being ripped away from each other. If they're not anchored, you know, absolutely incredibly strongly, the guy's going to fall apart right on the field. You see that? If I could home that in, you can see how it's anchored right into there. Anyway, that's enough. Okay, so right at the very beginning, he said they're going to go by people presenting videos with opinions and evidence. And here's going to be my opinion and my evidence. Now, he's talking with an expert about fascia and the mucosal surfaces that pretty much coat everything. It's the fascia. And when he says mucosa, I'm going with that being the interstitium, the fascia. So here he goes.
And occasionally, some of those people will die. Now, if you look at the people, usually there's no obvious difference. How would you predict that person was going to get a sniffle and that person would end up in hospital? And so we were able to say, well, wait a second, maybe there's a spectrum of responsiveness, a type of resilience And if we can make a poorly resilient person more resilient, then that's a very effective way of going forward and having a decent type of treatment without.
All right, now I agree with him 100%. It's all about your ability to fight back.
Vaccines and all the other bits and pieces that go along with them, or in conjunction with vaccines. Quite frankly, any vaccine would work much better if you've got a highly responsive, highly resilient immune system operating in the lungs. So we tested that, you might remember. We tested that in people who were essentially relatively normal people, and we found that about a quarter of people didn't turn on their immune system very effectively.
Okay, I just want to make my statement here. When it says it didn't turn it on to the immune system, in other words, the immune system didn't respond, which means to me the bacteria wasn't there to create the enzymes.
And also, we were able, work that we've been doing over 30 or 40 years, was that the way in which the airways get their protection, it's not making the antibody in the wall. So,
all right, this is where I believe he's wrong. The cells that are in the walls should have the ribosomes within inside of them to activate. They should already be there. So they're not necessarily making them right at that point, and they probably made in the gut 99% of them, but they float. They're called free floaters, so they go through the entire system and they end up lodging themselves inside of all the cells. You can have up to 1 million or 10 million in a single cell of these ribosomes, and they're the things that do the chemistry. That's the enzymes. So that's where I'm going. So let me just go back just a hair here, because I don't think he's exactly right on that. So here he goes again.
Their immune system very effectively. And also, we were able, work that we've been doing over 30 or 40 years, was that the way in which the airways get their protection, it's not making the antibody in the wall so much, it's taking the microbiome, which is in the airway, swallowing it and delivering it to these little factories in the wall of the small bowel.
All right, that's where I don't believe that's correct. I believe the enzymes that are created in the intestinal area in the gut, those enzymes, and also in all your secretion glands, those are the things that do the job. It's not that they're well, anyway.
It's an off-site system, leaving the lungs to just worry about one thing: putting oxygen into the body, taking carbon dioxide out. And we found that 1 in 4 people didn't do this very well. But if we gave them killed bacteria so that we loaded up this stimulation system.
All right, when he says that they gave them killed bacteria, they're giving them probably some kind of probiotic, killed bacteria. That's a lot of times that's what you're eating. So that we loaded up this stimulation system. Those are the things that are going to create the enzymes.
All of a sudden, they all behaved with high resilience. And that seemed to us a very sensible way of going forward. And it really only came as an idea as a result of COVID. Now, the microbiome people got in on the picture by saying, well, hey, wait a second, you've got an infection in the lung, but we're looking at the microbiome in the gut, and it's distorted. It's the type of microbiome that we recognise as causing disease. And when we looked at the people with COVID that got this changed microbiome, they were the ones who got very sick, and some of them died. And so
So obviously they didn't have the enzymes because they didn't have the bacteria. We need a bacterial database that says here's what should be in everybody's bacteria. If you don't have some of it, well then we got to replace it. It's basically as simple as that. But we need to make a very clean, consistent database, and it might have variations according to race and sex and all that business, age. But there should be some bacterial database. It's going to fall in line with the more swabs, the more fecal swabs, more, and feeding it into this data. We should create a very, very good database that would be the average, because there's going to be a standardized set of bacteria basically you're going to need to break down all the different things, the fats, the carbohydrates, the sugars, and create all the different products to save your life. And they have found these fecal matter transplants, which is you're just taking some of somebody else's poop and putting it in you, and it helps you. Just like you said, they put a bunch of bacteria in and pop them right up. It's basically the same thing.
Microbiome people and the mucosal immunology people started to come together, conceptually at least. We don't talk to each other or anything like that. That's not true. But conceptually, and we said, well, wait a second, maybe there is a single system where you've got one central component in the gut that's creating the immune system that goes to all the surfaces. And we sort of knew a bit about that before, but we didn't think about it as just a single system.
Well, that was my paper, 2015. That's what I wrote about. It's one giant system, fluid-filled highway, and that's exactly what he's talking about. Sounds like to me.
And maybe the microbiome is also part of a continuous system that can communicate up and down through the bloodstream with cells and molecules. And so we came up with...
Yes and no. Through the bloodstream to a degree. Primarily it's through the lymphatic system. And these bacteria and ribosomes are so small that they can fit through those little tiny lymph ducts, and they can all go through the blood-brain barrier too. So when he says in the bloodstream, yes, with the cells, yes, but primarily through the lymph nodes, through the lymph ducts and the lymph nodes. That's where they get cleaned up as they go through the lymph nodes.
The idea, this is a terrible name, and I'm awfully sorry, John, and you're the first person I've tried this out on, the Mucosal Immune Microbiome Protection System. Now, you have to have some...
Well, anyway, that's the new name.
Something that is trendy, and that's a MIMPS. So here you are, John. It's the first time I've talked to anyone about MIMPS.
You've heard it here first, everyone. This is the new way forward.
Last time you ever hear of it, but immediately everyone listening to this will say, wait a second, that makes sense. Yep. That if we're going to treat somebody with because this is what's happening now in diabetes and obesity, in inflammatory bowel disease, we're saying, well, we're going to treat the microbiome. Yep. But if you've got a system where the outcome of the microbiome is essentially the modal outcome, or the interaction between, on one hand, the pressure of the infection, the microbiome, and the pressure of the body's resistance, the immune response, then that sliding outcome in one way determines the resilience that we were talking about before. But on the other hand, it also says if we're going to get a better outcome, maybe we should have a dual approach to treatment. Treat the microbiome with everything from probiotics to fecal microbiome transplants, where you exchange that doesn't sound very pleasant, but it's highly effective in certain circumstances.
I think it's going to be highly effective in almost every circumstance. If you take into account it's the bacteria, primarily in the feces. However, there's another study out that I was just looking at that they're taking. I think it was 22 sites, something like that, in the body. Oral swab under your armpits, your in your groin area, your feet, and all kinds of. You have different bacterial communities living in all those places, and in every single organ in your body. There's a lot of work to do here, but we're starting to understand it, and this guy is talking exactly what I've been saying.
But combine it with boosting the mucosal immune response. And the second immediate outcome...
Just to be sure you understand, when he's talking about mucosal, that mucosal area is the interstitium. That's what the interstitium is. In the mucosal area, that he can call a mucosal area, but it's interstitium, fascia, same basic thing. All right. right, just so it's 100% clear what he's talking about. When he's talking about mucosa, it's nothing more than the fascia. Now, the mucosa is another name for mucous membrane. So the membrane is there, and it has all this little gooey stuff on the top. And mucosa lines the inside of the organs and cavities throughout your body that are exposed to particles from the outside. The mucous membrane lubricates and protects these organs and cavities from abrasive particles and all kinds of nasty stuff and bodily fluids, as well as invasive pathogens. You have more than 200 times more mucosa than skin. It's basically this interstitium that they're talking about now, making it your body's largest protective barrier, and it's called a new organ now. It's also involved in absorption, especially in the gastrointestinal tract. That's the lymph ducts, where it plays a role in digestion. Now they're talking about lubrication. Let me show you something. This is very, very cool. If you can see this. Let me see if you can see that. Synovial joints in humans, and that's nothing more than synovial fluid, sinus fluids, is 4 times slipperier than Teflon on Teflon. It's the slippiest thing there is. These are all different coefficients of friction, how something slides against something else. And that one there is frictionless. It's the most slippery thing there is, basically. So that's why you can glide so well. However, that stuff can set up in you. If you don't keep it moving, eventually it sort of glues up a little bit. And that's when you get really stiff. And that's what stretching and all that stuff is about. But in the gastrointestinal tract, it absorbs all of the fluids that are going back and forth. Some of it's coming in, some of it's being flushed out. And that's how you get your nutrition, and that's how you get your bacteria and your enzymes and all that stuff. And it's what is it composed of, and then they go through the different layers and the function of mucosa. It's in your immune system. That's just the barrier. And they called it mucus a while back, but it's the same thing. It's the fascia. And now they call it the interstitium. There's so many names for things. And if I showed you some just before, some guy's got a new name. It's got like 4 or 5 words in it. All right, so don't forget, I've shown vast amount of information that supports what I want to see done, which is a database. It should be done, and I think there may already be one, but I don't know how to access it. I don't know how to put things together. I just do research. But again, I think I showed this before. We need a universal database, all bacteria, and then we can get a good handle on what's going on, because there is nothing that happens in your body that isn't triggered by an enzyme. Not a single